Yes, Devanshi, where are other students? Are they joining? Hello, am I audible?
Yes, ma'am.
Yes, ma'am.
Okay, Devanshi, where are other students? Are they joining?
Ma'am, yes.
Okay, you were having class.
Ma'am, we actually had a test in the previous class, so I think some of them are still submitting.
Okay,
So,
okay, fine, fine. No issues, no issues. We'll
yeah.
wait. So, okay, to begin with, last lecture was about the symptoms that we discussed, practical basically I'm talking about, right? So, in symptoms, we discussed about the various types of the symptoms which are caused by the viruses and the plants, and how to identify and why it is important to study the symptoms and the viruses, symptoms by viruses in the plant, right? How does this helps in the diagnosis? And how this is the first steps towards the diagnosis of any plant virus. So, yes, can anyone tell me that why it is important to study symptoms caused by the viruses in the plants? What is their use? Yes, ma'am is here.
ma'am, it's important to study symptoms because then it will help to identify the disease later if needed, and it can also help us develop a proper treatment method against the virus by studying its
Yes.
symptoms.
Yeah, you're right. So, see, the first thing is that plants cannot talk themselves, right? So, they are not going to go to the doctor and tell that okay, these are the symptoms we are having, so please treat us. So, this is the first thing that they cannot say anything. It is us that we need to look into the symptoms and we need to look what kind of a pathogen or diseases they are, some pathogen they are causing diseases. And why it is important? Because given the economic importance of the pathogen or viruses, they cause economic losses, both qualitative and quantitative losses. So, in order to reduce those losses, the first step is to identify the pathogen or a disease, and then the step is for the management. So, in order to identify the pathogen, the first step is the visible symptoms that they cause on the plants. So, on the basis of certain symptoms, we can say that okay, this could be this virus, or this virus could belong to this particular group. Or if not at all, we can say that this is not a fungal pathogen, this is the virus, right? So, initial diagnosis
Your voice is very
is this clear for everyone, only for few students? Yes, students, answer.
Ma'am, you're not audible.
And now,
Now it's a little better.
Okay, and now, is it fine now? Hello, is it better?
No, ma'am. It's
Better. Others, please, because sometimes the disturbance is at the end of the students, one or two students. So others, please. Other than Yukta, others, yes. Ayushi, can you hear me? Sam, Devanshi, Janvi.
ma'am. Yes.
Yes, ma'am.
So Yukta, please check your network because others can hear me and there is some issue at your end. Then please just try to leave the meeting and join again.
Yes.
Try to do that. So that's why I was telling that symptoms is very important. So my another question is that how we can just by looking at the symptoms tell whether it is a deficiency, nutrient deficiency symptom or it is a viral symptom. Yes, who will answer this question? Because nutrient deficiency symptoms are usually similar, which are caused by the viruses. So how by just looking at the field in different plants, we can say that this is not a viral, this is a nutrient deficiency. Yes, yes, Sam, answer the question.
Ma'am, I'm guessing that if there is a nutrient deficiency, it would be visible in like the whole field of plants, whereas
Yes,
virus
yes.
is in specific.
Yes, you're right. Fine. Yes, that's correct answer. That if there is a nutrient deficiency, it will be visible on the entire lot in the plant. So it's not that it will be visible only in a few plants, because if it's a nutrient deficiency, that means the whole field has a deficiency of that particular nutrient. Whereas if we talk about the viruses, the symptoms shown by the viruses will not be in all the plants. Maybe two, three plants in patches, maybe 10 plants in a patches, or maybe just one plant in a patches. So that is the major difference. So by looking the initial types of the symptom in the field, we can say that, okay, this is not a virus symptom, this could be the nutrient deficiency. So we should further go for some nutrient testing in the soil, and then after that we can go for the virus testing. Yes, right. Yes, Ishita, can you tell me about the common virus symptoms that are caused by the viruses in the plant, common symptoms by which we can identify them?
What, ma'am?
Ishita, Ishita, can you hear me? What are the common symptoms caused by the viruses in the plants? Rupal, Nandini, no? Nehal, I guess this is the second month we are studying viruses, and from day one I'm telling you about the
Ma'am, you
virus.
were audible, but someone removed me from the meeting. I don't know.
Okay, now I'm audible.
Yes, ma'am.
Okay, so what are the different symptoms caused by the common symptoms? Just one student will tell one symptom caused by the viruses in the plant.
Ma'am, leaf curling.
Leaf curling, yes, good. Another one, Shakti,
Oh, yellowing of leaves.
yes, Yuvraj,
I'm fading color and unwanted aberration.
change visions, okay, fine. Yes, other common symptoms, mosaic, stunting, mottling, vein banding, vein clearing. So these are the typical symptoms which are caused by the viruses, right? So I hope you all have written the first experiment in the field in your practical file. So if not, then please complete your practical file, write the first experiment as the symptoms or the identification of the symptoms, right? Okay, then coming to the internal symptoms. So it's histological changes. So what are the common histological changes which are caused by the virus infection in the plant? Can anyone tell me? Cytological changes or histological changes that we call. You remember we talked about the inclusion bodies. With the virus infection, leads certain group of viruses, they form inclusion bodies, certain different types. There are crystalline inclusion bodies, there are thin wheel inclusion bodies, there are, yes, nuclear inclusion bodies. Yes, Sam?
Ma'am, hypoplasia, hypertrophy, and necrosis.
Hy- Necrosis, yes, necrosis. And it will be under the microscope. See, visual symptoms are, yes, we can see with the eyes, but if we want to check under the microscope, so different kind of changes are basically the major one is the inclusion body formation, by which we can identify that particular virus is infecting a particular plant, right? So first practical, please complete and submit before the next practical that we have. And today's practical also you will complete. So today we are going to talk about the mechanical inoculation or the mechanical transmission of the viruses. So this is a very common practical or experiment that we do in the lab. It is very easy. We select some host plant, we inoculate the virus by making inoculum preparation, and then after some time we see for the symptom development in the plant. And after some time, when we see certain kind of a symptom, then we take isolate RNA or DNA depending upon the virus, and then by PCR or sometime by ELISA also we can confirm that the virus has been transmitted, right? So today's practical would be the transmission, mechanical transmission. So simplest, in a simplest term, this figure is telling you about the virus transmission. So we take an infected sample. It could be a leaf, it could be root part also sometimes, it could be a bark part also sometimes, it could be your fruit also. So depending upon the virus concentration in which part is the higher, we take that portion basically, and then we extract it in a grinding buffer. So that means we are going to crush it, we are going to crushing it in a some kind of extraction buffers are there that we'll discuss later. And after that, with the help of just using a finger tape maybe, or just by the cotton gauze or a cotton swab, we can just lightly will dab, it will abrase the leaf or a part of the leaf where we want to inoculate. That means we'll do certain kind of a wound abrasion, leaf abrasion is being done. So that because viruses, in order to enter, they need a certain kind of a wound. So just by using certain kind of a carborundum powder comes, they are very fine powder. So gently rubbing that powder, we can abrase the leaf, and then we can inoculate the virus. And then after some time, we'll observe for the symptom formation. And when the symptoms are formed, maybe after 1 week interval or after the 2 week interval, the symptom will be formed. And then we'll check whether the virus is present by the ELISA or a PCR. Then we can say that, oh, okay, after confirmation by ELISA or PCR only, we'll say that this particular uh, virus has been purified. So this is the common method of obtaining a pure culture of the virus in case of the plant viruses, right? So as we have discussed transmission or the transmission methods already in the theory. So mechanical transmission means the transfer of pathogen from an infected host or a contaminated substrate to a susceptible host, where the biological association between the pathogen and the vector is not necessary. So this means in this kind of a transmission, we don't require any kind of a vector. It can be done mechanically without the vector also. And usually, why we do this kind of a mechanical transmission? First, if we need to purify a virus, in that case, we need to do this. Or if we need to see, viral infected lymph samples will not be available throughout the year, right? When a particular season is there, that plant, the virus is there for some time, it will be available. So if off the season we want to do certain experimental works, we want to RNA isolation or a DNA isolation, or we need a sample for the further any other kind of a study. So they will not be available throughout the year. So we can maintain these plant viruses in a suitable host, and then from that host we can use it whenever we want, right? So for that purpose also it is required, and it is usually done by the researchers. Then source and preparation of inoculum. So the virus inoculum for a SAP transmission. We call it as a SAP transmission also. It can be obtained from the various parts of the infected donor plants. But usually younger leaves are preferred because it is generally found that the younger leaves they have a higher concentration of the virus than the older mutations. Reason is there could be several reasons because in the older leaves or older plants there are secondary metabolites which are being formed. There are certain compounds, phenolics or certain other compounds are being formed due to which the virus concentration becomes less. So leaves usually which are showing virus symptoms are chosen and then particular portion of the leaf is being taken, or sometimes we can take it also depends upon the different viruses. If the virus concentration is more in the root part, then we take the root part also for the preparing virus inoculum. So in case of few viruses such as soil-borne tobacco necrosis virus, the roots of the infected plants may be a better source of inoculum content. So in that case, we take leaf. Instead of taking leaves, we go for the roots and we crush it and prepare the inoculum. So after selection of a suitable piece of an infected leaf or a tissue, then we prepare the inoculum by grinding of the tissue in a chilled mortar and pestle. So usually this kind of a work is done in a cold room. The temperature preferred is less than 0 degree Celsius so that viruses do not degrade. Viruses, each virus, they have a thermal inactivation point, which is called a PIP also. So it is usually lesser, towards the lesser side. So at higher temperature, they tend to degrade. So even if we try to inoculate and virus is degraded, the transmission will not take place. So we recommend that this kind of experiment should be done in a cold room or in a colder conditions. So whatever the means of grinding the tissue, however, various host metabolites and cellular debris will be released. So once we are grinding, number of metabolites and other cellular debris, they will be released along with the virus, right? So what happens that some of these compounds they can cause the virus inactivation also, and they can affect the infectivity also. So it is usually to grind the leaf in a suitable buffer at a low temperature to minimize the loss of virus infectivity. So infected tissues are often ground in a zero point 0.1 molar uh, potassium phosphate buffer, which pH should be around 7 to 7.5. So if the sap of the donor plant is particularly acidic, then the buffer should be used at a pH of 8 to 8.5. But then we should also remember that excessively alkaline condition can also cause the breakdown of the virus, right? So pH should be properly maintained. Then we also add certain reducing agents to the phosphate solution. So these kind of reducing agents, they prevent the oxidation, hence the loss of virus infectivity is also being prevented. So sometimes a solution of 1% dipotassium hydrogen phosphate containing 0.1% sodium sulfide is good for isolating and transmitting many viruses. Also, see, these kind of procedures are usually standardized for different viruses, right? There is one common protocol in which we might require certain changes according to the virus that we are using for transmission. So usually for different viruses, there could be different components सच एज अगर फिनोलिक कंपोनेंट्स ज़्यादा है किसी टिश्यू में तो हम उसमें रिड्यूसिंग एजेंट डालेंगे वी कैन ऐड सम अदर एजेंट्स आल्सो राइट इन ऑर्डर टू इनएक्टिवेट दिस काइंड ऑफ कंपोनेंट्स सो depend upon the kind of the plant which we are isolating the virus and also upon the virus that we are isolating. For example, the addition of PVP. PVP is polyvinyl pyrrolidine to the extraction buffer is frequently used to protect the virus against phenols in a host such as rosacea species. So the plants in a rosacea species, they have a very high phenolic content, such as the stone fruits also and foam fruits also, they have a very high phenolic content. So when we try to isolate even RNA isolation, so these phenols, they inhibit the process. So in order to prevent that, we add this PDP in that particular extraction buffer so that they don't hinder any process, right? So once the infected tissues, they have been homogenized, the sap inoculum may be used directly or in order to just filter the debris, to remove the debris, certain post material, we can do the filtration also, right? With the cotton gauze to remove the larger debris. So if then there is another situation that if the inoculum is not used immediately, or if a larger number of the test plants are to be inoculated, then we need to keep it in a cool ice bucket. So usually what happens? Glass houses are away from the labs. So usually inoculum is being prepared in the lab, and then we go to the glass houses outside the field. So that inoculum basically in a vessel motor, we take it in a bucket, ice bucket, cool bucket, properly covered, so that the temperature or a lower temperature is being maintained. Then the next step is the inoculation. So during SAP transmission, virus can only enter through the wound. We know that either through the vector also cause wound and then enter, or through certain kind of a wound is in the plant. So we need to make some kind of a wound, minor wound in the plant, so that a virus can get an entry, right? So fine abrasive powder such as 300 to 600 mesh carborundum. It's a silicon carbide or a silite, which is a crushed diatomaceous earth. These are commonly used to produce the entry. So very small amount we need this kind of a powder, and abrasion is made, right? So these powders are usually blown onto the leaf surface of the test plant using the throat powder spray. Certain kind of a spray can be done prior to inoculation. So these powder, they will settle on the plant and they will cause minor abrasions, right? It is covered with the light layer of abrasive. So after, when we try to inoculate the sap, the finger or with the cotton gauze, abrasion will be produced on the leaf surface. So alternatively, the abrasive can be mixed with the sap inoculum also. So we can mix the abrasion with the sap inoculum also, and then we can inoculate the plant. Then methods of applying the inoculum to the leaf tend to vary from lab to lab. And some workers, they apply the inoculum with their fingers, gently rubbing the wet finger over the whole upper leaf surface, test leaf, whatever leaf we are choosing, maybe 3 leaves, 4 leaves, right? So if a finger is used, then hand must be thoroughly decontaminated before different inoculations. But sometimes the workers, they use the cotton gauze or a sponge or a cotton balls, also small cotton balls, to gently rub the leaves with the inoculum. So application of the inoculum by spraying, usually it does not result in a virus infection, but it is being used for adding an abrasive to the inoculum before spraying. That can be done. Then the virus, it enters the plant tissue through the broken leaf hair cells or other wounds which we have caused using the abrasive material in the epidermis. So although most viruses can be mechanically inoculated in the sap by these methods, infection will only occur if virus is able to multiply within the epidermal cell of the recipient host plant. So definitely, if it is multiplying further, then only certain kind of a symptoms, maybe local symptoms or a systemic symptoms, will be produced in the plant. If the infection process is successful, the virus will multiply and it will spread to the other cells also. So local lesion can form and virus can be. Local lesions are basically formed at the point of the entry. Certain pinhead spots, black color spots are formed on the leaf, and then it spread to the vascular system, to the systemically throughout the plant. So there are certain factors which affect the mechanical transmission. There are inhibitors and inactivators. So loss of inoculum activity, which are usually caused by the chemicals which are being released from the donor plant during the homogenization. Yet when the extraction is being done, is a major problem in the mechanical transmission from some of the donors. So in considering loss of virus infectivity in SAP homogenates, it is important to distinguish between chemicals that inhibit and those that actually inactivate the virus. So some donor species, they have a very strong inhibitors, due to which it is sometimes impossible to SAP transfer viruses to other species, right? Even when the recipient host is susceptible to that virus. So such inhibitors include enzymes and certain kind of a polysaccharides, and they are common in the leaf sap of such plants such as Chenopodium, Dianthus, and other plants that are being used. So action of some inhibitors can be overcome by diluting the sap samples to a level in which they become ineffective. But then we need to look into the virus concentration also, कि उसकी concentration इतनी कम ना हो जाए कि virus infect ही ना कर पाए. Virus load is too less to infect the particular plant. So action of other inhibitor can be overcome by addition of certain chemicals such as bentonite to the grinding buffer. So certain reducing agents can be used along with the grinding buffer grinding buffer, which can inactivate these kind of compounds. So, for example, in nature, many woody plants contain virus inactivators such as tannins and oxidases, and these can be rendered ineffective by addition of a high pH buffers, reducing agents, gelating agents, or protein binders to the grinding medium. So, alternatively, workers may try to avoid donor host plant known to contain high level of inactivators. Sometimes it is possible to use the young leaves of a woody host because जो young leaves होते हैं, they have a lesser concentration of these phenolics or these compounds. So we try to use those young leaves. Now, what kind of a host plant we need to choose? How we decide that this is the host plant which is suitable for our study. So for most hosts, the leaves are the most susceptible and convenient part of the test plant to inoculate the virus. For example, species such as Phaseolus vulgaris, which is a beans, the primary leaves are mostly susceptible and the later developing trifoliate leaves. Then in some species of Cucurbitaceae, the seedling or cotyledon stage are more susceptible. So we need to inoculate leaves or plants in those stages only, not at the higher stages. The choice of the recipient host plant to be used for mechanical transmission depend on the virus and experimental objectives. So usually there are different host plants which are very commonly used for the purification purposes. It also depends upon whether we want to store the virus for a long-term propagation of a virus, or it is just a rapid multiplication for the purification purposes, right? So what kind of experiment we want to do? So on that also the host selection of the host depend. So frequently, the common lab host plants such as Inoculation species, Inoculation tabacum, Inoculation banthanana, Ginocodium album, Ginocodium species, Phaseolus vulgaris beans, they may be suitable for these purposes. These are the common host plants which are being used and virus is being maintained on these host plants. So these host plants are basically infected by any kind of the plant virus. They are host to many viruses. That's why they can be host type. In other cases, some viruses they may have a narrow host range and infection may be restricted to one family or a genus of the plant. So in those cases, we need to look for those plants only. So these are the common plants such as Inoculation and tabacum. And here you can see the cucumber mosaic virus is mechanically transmitted to the leaves, younger leaves when they were young, starting leaves, right? And the symptoms are being produced. Then there is a Inoculation and again, which is commonly used. We have Phaseolus vulgaris, which is your beans, and then Chenopodium also. So these are the common host plants which are being recommended for the virus purification or the virus maintenance in case of the plant viruses. So this is the phosphate buffer, which is a standard buffer. pH can vary with the type of the virus or the host is required, but this is the standard buffer which is being used. We can add polyvinyl pyridine or certain other agents based on the properties of the virus and the host from which we are preparing an inoculum. Oh, okay, protocol. So this is kind of a detailed protocol that we go for. Okay, so this is the process basically, right? So first is we select a suitable test farm, right? After that, we make an inoculum by crushing virus infected. It could be leaves, it could be bugs, few fruit viruses such as stem eating or bloating viruses. Their concentration is more in the bark of the trees. So we can isolate them from that part also, or a root in a suitable extraction buffer, right? Then we select 3 to 4 leaves for inoculation, younger leaves. Prior to that, we grow those plants, basically Nicotiana tabacum or a benthamania, any plant. Through seeds, we grow them in a glasshouse condition, right? So they are grown. It's not that in the wild these plants are grown and we just inoculate them. No, the seeds are available and we use those seeds for growing these kind of a plant, and we make sure that these seeds are free from these kind of pathogens already, right? Then we use certain kind of a carborundum powder for the abrasion. We can use fingers to abrase the leaves along with the inoculum, or we can just spray the abrasive material on the leaves, and after that with the help of a cotton or a finger we can spread the inoculum very gently. Then after inoculum, sometimes we wash the leaves also to remove the excess of the inoculum from the leaves. And then after 1 week time, we can start observing the symptoms which are appearing on the these glasshouse plants which are being mechanically transmitted by viruses. And once we see certain kind of symptoms, now we again need to be sure that this is the actually virus that we transmitted. So if we have antibodies available against these viruses, we can go for ELISA testing, or we can go for a PCR or a RT-PCR depending upon the DNA or RNA virus to confirm the presence of the virus or the transmission of the virus to a plant. And once it is being confirmed, then we can designate it as a pure culture that now we have the pure culture of this particular virus and it can be used for the various other kind of this lab studies or this is the generalized procedure that we discussed, and it could be modified when we are working with different viruses or different hosts. So it can vary, right? So okay, so the experiment that you all will write will be, it is the experiment to explain mechanical transmission of cucumber mosaic virus from cucurbitaceae host to Nicotiana tabacum plant. So I'm not giving you the procedures. I've told you the generalized procedure. Now you will write yourself an experiment, right? That how you will go for the transmission of cucumber mosaic virus, maybe from the cucumber plant only to the Nicotiana tabacum plant. What will be the steps? What will be the buffer you will prepare? How you prepare the inoculums and how you will transmit it. So that will be your practical for the mechanical transmission of the CMV from cucurbitaceae host to the Nicotiana tabacum plant. If any clarification required, please ask me right now. No, it's clear? Fine. So next week, I should have the notebooks which contain your PDF files submitted, which contains both experiment 1 and experiment 2 also, right? So with that, I'll be checking your practical notebooks by next week. It should be submitted in the Teams itself. You can submit it in the file section. You can just name yourself and 2 experiments together. Don't submit separately, right? So, okay, so mechanical transmission part was the smaller one. I have one video to show you all. Let's see if I can play that video, share my screen with you, right? Is it visible, audible or visible both? Students, is it audible and visible both or not?
Ma'am, it's visible, but I don't think it's audible.
Just a moment, maybe I'll have to, just a moment, I'll have to use the computer audio, right? Just a moment. Is it audible now?
Ma'am.
Like this. So it's a very light curve of this very fine mesh, very fine magic band like around it. You can't build now, just prepare that. Use that.
Is it audible, students, or not?
Yes, ma'am.
Is it
Yes,
audible
ma'am.
now or not?
Yes, ma'am.
And that's enough inoculum to apply to leaves. Then we simply wipe the leaves 3 to 4 times with the inoculum. We inoculate up to 3 leaves, which are expanded. This is we apply inoculum to whichever plants we want to inoculate. So what we have is just a light coating of liquid inoculum on the leaf, and that rapidly dries off. But after the inoculum has been applied, the virus enters a number of cells and uncoats and starts the replication cycle. After about, we first of all then have to make sure that we label the plants with the virus and the date, so we know how long we should come back to check the symptoms. Symptoms usually take, we have 2 types of symptoms. We have some which are local symptoms, which can appear in 3 or 4 days. And here is an example. When a virus is not mechanically transmissible, we can still demonstrate that it's transmitted. We can transmit these symptoms using a method called abstract grafting. Grafting is a method where we take a piece of tissue from an infected plant and apply it to a susceptible other plants, the same species, and we cut tissue directly from the cambium of the tissue from the infected plant and match up with the cambium of the recipient plant. And then the virus can be translocated across from that patch into the recipient plant and infect from that grafting site. So what I'm showing here is the virus source of this plant is now showing mosaic symptoms. I'm now going to take a patch of tissue from the stem of this plant. So I'm going to cut into the vascular bundle, vascular ring. I'm now going to cut down parallel with that, and then cut at an angle a small patch. So we have now a patch of tissue like this, which has a little notch at the bottom. All we have to do now is take the recipient plant, prepare a place on the stem about the same diameter. We just have to remove a few leaves so that we have a good feeling here. So I now simply cut a piece of tissue out from the recipient plant with approximately the same dimensions as the piece I've taken from the source plant. I can discard that and then place that piece of tissue from the infected plant there. That is often held by a capillarity. There's enough moisture left there to hold that piece of tissue there. And then I tie it with a waterproof tape. We can use various kinds of tape, plastic tapes and rubber strip also are very useful. And we simply wrap the tape around the stem, cover the patch so that the patch is held firmly up against the vascular bundles, the cambium, parts of the cambium of the recipient plant. And then in a few days, we will see symptoms appearing on the top of the recipient plant as a result of the virus translocating from this inoculation site to the healthy recipient plant. So here we've tied it off. The patch is now inside there. It's not become dry because the virus can now translocate from infected cells into adjacent healthy cells of this new host, start to multiply, and then eventually into the phloem, be translocated up and down the stem of the plant. So with a plant like this, we can normally trim it off so that when the new growth comes from the axils here, we will look for symptoms on this new growth. Now, this is a technique we can use with plants which have, or dicotyledonous plants which have vascular bundles. But unfortunately, we can't use this technique on a complete class where we have covered elimination. But this is a method here we can transmit viruses which are contained in certain tissue sacs of the blood. We don't actually get into the sac and extract the sac. So it's a better method for ensuring that we get transmission of the symptom when we cannot get mechanical inoculation. So these are two standard methods we use for transmitting viruses in the virus process. Firstly, mechanical inoculation. Secondly, grafting transmission. In this case, we call it patchback transmission because we're taking the patchback from the donor placed on the recipient. There are other methods which you will see in notebooks, in textbooks, and literature, showing different methods for doing this because it's a standard method we use and it simply allows us to confirm whether a virus like symptoms is transmissible. The principle about diagnosis of viruses is that we need to show that we can transmit the symptom from
Hello, am I audible? Hello, am I audible, students?
Yes.
Fine. So this was the simplest technique that was explained, and they just a moment. Yeah. So first was the sap inoculation, and in second method it was described about the graft transmission basically. So graft transmission is also a very commonly used method in plants which are grown through the grafting, right? Sometimes the rootstock that we use is of a different variety and the main plant is of different variety. For example, in case of apple and stone fruits, this is a very common technique which is being followed. And why this is done? Because if we use the same rootstock of a tree which is giving fruits, sometimes it is very weak. The rootstock, the original root system is a very weak. So we use the rootstock from a stronger variety or from some wild variety which has a strong, which is more strong as compared to the rootstock that we have. So that is why we go for a grafting. So rootstock that we use is from the wild species or some of the wild relative, and that on that we use our tree in which the fruit yield or which is the fruit that we get on the main tree is a more, right? So rootstock from a different variety and the main tree which yields fruit is from a different variety. So graft transmission of viruses is very common through this method. If rootstock is infected by certain kind of a viruses, then the whole tree eventually will become affected by the viruses. So we need to be very sure that the rootstock and other part that we are using in a grafting, right, they should be free from any kind of a pathogen or any kind of the viruses. So this is why the certification method, as from the beginning, I'm telling you that in the management, we lay emphasis on the certification of the starting material, that they should be free from any kind of pathogen. So these are the two methods which are very commonly used. So in your practical file, you'll only write the mechanical inoculation by sap transmission, not the graft transmission, right? So I've given you the name, title of the experiment in your own word. Please, it should not be copy pasted from one notebook. No one will share their notebook with anyone else. I want to see what exactly how you prepare experiment individually. So मुझे सबका अलग दिखना चाहिए, किसी का भी same नहीं होना चाहिए. Please make sure that write it in your own simple words. No copy pasting from the book or a Google anywhere. मैंने जो procedure दिया है उसको use करो और उसको fit करो उसमें CMV को transmit करने में. It should not be copy pasted from anywhere, right? Make sure that, right? And then we have two classes, right? Today we have two practical. So let's take attendance for this class and then we'll take a 10 minutes break and then we'll come for the next session. Quickly switch on your cameras for attendance. Adish Mishra.
Yes, ma'am.
Adish के बाद है अथोइबा.
Hello, ma'am.
Yes, Athorva, I can see you. I can't hear you so clearly, but Ayushi?
Present, ma'am.
Sam?
Present, ma'am.
Yeah. Devanshi?
Present, ma'am.
Ishita?
Present, ma'am.
Janvi?
Present, ma'am.
Kshatij? Kshatij, where are you? Manaswani?
Present, ma'am.
Yeah. Namrita?
Present, ma'am.
Nandini? Nehal?
Present, ma'am.
Rupal?
Present, ma'am.
Sahiba?
Present, ma'am.
Sanjana?
Present, ma'am.
Shakti?
Present, ma'am.
Shivani? Tanvi?
Present, ma'am.
Tridiv?
Present, ma'am.
Vivek? Yukta? Yuvraj?
Present, ma'am.
Yeah, Yukta and Yuvraj present. Yukta, some problem at your end. Okay, so let's meet at. Yes,
Ma'am, I'm sorry.
yes.
My Teams is not working. I'm present.
Okay, fine. Fine, Nandini. So 12:15, we'll again meet for the next part of the practical, right? Quickly, we'll come after the break. Thank you so much.